Greetings, I'm Yvonne, staff for Science for the Public, and I welcome you to Contemporary Science Issues and Innovations. As global warming accelerates, a major concern is how organisms will or will not be able to adapt to climate extremes. As part of the answer lies in DNA. Our guest today was one of the pioneers in the study of genes and adaptation to climate change and the implications of that connection for both biology and medical science. William Detrich is Emeritus Professor of Marine and Environmental Science at Northeastern University. For decades at Palmer Station and McMurdo Station in Antarctica, Dr. Detrich has directed innovative research in the extreme, unique adaptations of the Antarctic icefish, especially its remarkable white antifreeze blood and its skeletal change that ensured survival in the extreme cold of the Southern Ocean. In recognition of his outstanding discoveries, an Antarctic island near Palmer Station has been named Detrich Island in his honor, and we are honored to welcome him to Contemporary Science. Thank you, sir, for joining us.
Well, thank you very much, Yvonne. It's great to return to your program.
Yes, and a pleasure to have you back after several years now. And so I'm going to ask you to start with a little bit of background of what happened to that climate in the Antarctic, whatever it was 40 million years ago or whatever, that shift that caused this need to adapt or die, please.
Well, that's a very interesting question, and I would like to frame it in the context of geological time scale, as opposed to changes that are now occurring in human lifespans. Mm-hmm. So about 40 million years ago, the ocean around Antarctica was actually pretty temperate. And if you look in the fossil record from that time, what you see is a great diversity of fishes. Many different fish species, many different groups were present. Well, the temperature then was about 18 degrees centigrade, kind of like summer temperature in Massachusetts Bay. And over time, it began to cool. The reason for this cooling was twofold. First, the tectonic plates around on which Antarctica and South America and Australia sit, they began to move, and it opened up a southern ocean surrounding Antarctica that isolated it from the northern oceanic regions. At the same time, carbon dioxide in the atmosphere was declining. And these two events are thought to have led to the isolation of the Southern Ocean and its cooling over 40 million years from 18 degrees centigrade down to -2 degrees centigrade, the freezing point of natural seawater. So the fish and other organisms in the Southern Ocean have about 40 million years to adapt. If we concentrate on the fish, what one sees is that as time progresses towards the present, most fish groups disappear. They apparently chose to live in warmer waters to the north. They became locally extinct in the Antarctic. They didn't go extinct, but these groups were no longer represented. Well, there was one group that did stay behind. There's actually several groups that stayed behind, and one is the group of fish that I study. And they apparently had the genetic right stuff to become adapted to living in the cold.
So they stayed put. What were the changes? Their physical changes.
Well, I'd like to tell you about two of the major changes that occurred. The fish, called notothenioids. I'll just refer to them as
Yeah.
Antarctic fish in this context. But these fish had a buoyancy adaptation that allowed them to migrate into the water column. Remember, most of their competitors had disappeared.
Yeah.
There was a lot of food available, but the original notothenioids were negatively buoyant. They had to use a lot of energy to get into the water column. This adaptation of becoming more closer to neutrally buoyant actually occurred prior to glaciation in the Antarctic. This is an example of what we call historical contingency. There was enough genetic variation in the notothenioid population that natural selection was able to choose for more buoyant bodies. And the fish then began to occupy ecological niches where there was a lot of food.
Right. Now, yeah, their changes were in the blood, and you mentioned the skeletal change.
Right.
Now, so they became lighter, profoundly lighter, right, in skeleton, and the blood.
The blood is the other trait that I wanted to describe to you. As the Southern Ocean was cooling, cold water dissolves more oxygen than warm water does. And it reached a point where there was sufficient oxygen present in the water that even the red-blooded species stopped using their red blood cells so much to transfer oxygen. And one of the 8 groups of these fishes, the ice fishes, actually lost the genetic material necessary to make hemoglobin, the transporter of oxygen in red blood cells. Why did this happen? Well, if you take a red-blooded relative of the ice fish and expose it to carbon monoxide, poison its hemoglobin, the fish doesn't die. Unlike us, if we have 40% of our hemoglobin poisoned with carbon monoxide, we die. But the Antarctic species had become less reliant on their red blood cells because oxygen was plentiful, physically dissolved in their blood fluid.
Oh, how interesting. And then, so how did they get oxygen if they lost the red blood cells?
Well, the ice fishes lost red blood cells because they weren't under selective pressure. They are simply living on oxygen that it passes through their gills into their blood, is physically dissolved in their blood fluid, and that blood fluid circulates to the tissues, and the ice fishes obtain enough oxygen to do their metabolism.
That's really interesting. That's quite dramatic, isn't it? That so they could navigate easier because they lightened their skeletal very significantly, right? And then they were able to obtain oxygen more expediently, I guess, because of this change from having regular hemoglobin to this.
Yes.
I call antifreeze, or I think you called it antifreeze at one time.
Antifreeze is a blood protein that prevents freezing. We're talking about red blood cells and their loss. The red blood cells were no longer under selective pressure because, or they were at least under reduced selective pressure, and the ice fishes were able to discard them altogether.
Before we go to the actual DNA pioneering work, were there a lot of other species, a lot of other organisms in the water that made a similar change?
No, the ice fishes are one of 8 families of the group that I study. The other 7 families retain their red blood cells. Nevertheless, they're very closely related to each other. It's
Oh,
only the icefish is one of the 8 families that lost the ability to make red blood cells.
I ask that because just recently I don't know, there was a big discovery of these icefish nests, which just shocked people. And so it's been in the news that they seem to have done extremely well. They're taking over the ocean floor.
This is a stunning discovery. In
Yeah.
fact, it tells us how little we actually know about the Southern Ocean. So there's a discovery of, I think it's a 240 square kilometer
It's
section
amazing.
of the Weddell Sea in which one species of icefish is nesting side by side, and there are millions of individuals guarding their eggs in this region. It was absolutely stunning, remarkable, crazy. I don't know how to describe it.
It surprised you too, in other words.
Oh, yes.
A lot.
Big time. Yes. Yeah.
That is really amazing. Well, I would like to now go into your DNA part there. You got into this before people could even get started on working on it. There's quite an interesting story about you on that. Could you please tell us that it started out with the big genomics study, but then it couldn't be applied, I guess, directly, but you were thinking when you started looking at the ice fish, apparently you were thinking DNA, and you did something about it. Could you tell us that story?
Well, sure. Thank you very much for your kind remarks. When I was beginning my career, I worked on proteins, and I did study the proteins of Antarctic fish for some time. But at the same time, when I was in graduate school, suddenly people were able to clone DNA and study segments of DNA from organisms of interest. Well, this wasn't being applied in the Antarctic. And the reasons for that are manifold. The Antarctic is really difficult to get to. It's hard to obtain specimens. It's difficult to prepare DNA. Studies in the field in the 1970s and 80s were largely ecological in nature. What I realized was that we could now, provided we could satisfy all those issues, we could apply molecular biology and genome sequencing to these species to learn about their unique and remarkable traits. So that's what I set out to do. I'm not alone in this effort. Other investigators have done the same. But I was among the first to seize upon DNA as an additional means to understand these organisms and their interesting and remarkable ecology.
And so you collected a bunch of samples, is that correct? And kept them and waited until the opportunity arrived to start doing the analyses?
Exactly. So what we originally did was use molecular techniques to clone individual genes. So for instance, I studied a gene that codes for a protein that's involved in mitosis and the separation of chromosomes and so on. Well, around about 2000 or so, the human genome had become a major target of biomedicine. The idea being, if we understood the human genome and its genes and how they interact, we would be better able to study disease. Well, I reasoned that if we were able to begin genome sequencing of Antarctic fish species and other species in the Southern Ocean, we would begin to be able to understand their unique adaptations to the environment. And what's more, we could actually make use of some of the unique characteristics of the Antarctic fish to solve biomedical problems. So that's the other aspect of the work that you mentioned in your introduction that we'll talk about.
Yes, I'm right on that, as a matter of fact. So first of all, were you able to identify a genetic advantage there for this fish that it could quickly adapt, or it seemed like a fairly quick adaptation?
Yes, well,
It was a DNA advantage.
yes. In fact, the development or the evolution of the neutral buoyancy characteristic is the major trait that defines the nototheniod fishes. And based on our work using genomes, we were able to establish that the genetic variation that allowed this trait to evolve actually occurred prior to glaciation in the Antarctic, which is a striking finding. That you kind of think that it gets cold, so the organism evolves in response. Well, this is a situation in which there was what some people term pre-adaptation. The genetic diversity was there that could then lead to this useful trait.
Oh, so it was sort of in process, and despite climate change, it just happened to be a good accident for them.
It was, yes. Many things are accidental.
Yeah, as you would know, right? But that was, I had thought when I was reading about it that was a specific, very specific kind of change. Well, Dr. Detrich, if they could change make this kind of shift, if they have that DNA advantage to shift to extreme cold, would you predict that creatures have the ability to shift to extreme heat as we're undergoing now? Does it work both ways if you have an advantage DNA wise?
The potential is there for it to work both ways. But there are going to be winners in evolutionary terms, and there are going to be losers.
Yes.
So the situation is that the Southern Ocean is now warming, and it's warming by a degree every 50 years.
Much faster. Is that the case than previous changes?
So, when the Southern Ocean was cooling, it was cooling at a fraction of a degree centigrade per million years.
Yeah.
Now the ocean is in the process of warming at a rate that is, at least along the Antarctic Peninsula, about 1 degree in 50 years. So here's the problem. It's occurring orders of magnitude more rapidly than it did historically over geological time.
Right.
But I still think there is the possibility that some of the organisms will be able to will have sufficient genetic variation that they can evolve and withstand or sustain the species at warmer temperatures.
Okay, so there's an advantage just to change, sort of. There's that they have an advantage, the DNA makeup that would allow a better change, but there are many difficulties with this particular climate shift. Okay.
Right. They provided the species have sufficient genetic variation, it's possible to imagine that some of them will survive. In particular, what I want to point out is that as the Southern Ocean warms, around the continent, there are likely to be refuges that remain cold and don't warm as rapidly as the whole body of water. So these refuges would provide places where the fish could be present, occupy, still cold, have the chance to evolve in perhaps a more slow warming environment, kind of like micro environments.
Yes, yes. Right. So they can hole up there for the time And some others will be able to just move on. It's just that the ocean warming is particularly scary. It seems like right now it's very rapid and it doesn't reverse and so on. But in any case, thank you for that. Now I'd like to know about the applications of your work. You were really early on that too. The applications to the medical field. And I think especially around this skeletal loss. But anyway, what was that all about? Because I don't know of anybody else that got into that.
Well, if you're an evolutionary biologist, what you realize is that the fitness of a particular trait is context dependent. It depends upon the environment in which you are. So for the notothenoid fish swimming in the Southern Ocean, being neutrally buoyant is an advantage. Having a reduced skeleton is an advantage. For humans, in a one gravity environment where we're trying to maintain an upright posture and so on, a reduction in bone density and so on is a disease. I mean, it predisposes to a disease. So osteopenia, which is reduced bone mineral density, predisposes to osteoporosis, which is fracture of brittle bones.
Right, and there's a lot of that.
This is a major cause of mortality in humans.
Yes.
Low bone mineral density is an advantage to the Antarctic fish. So this points out the fact that traits that are advantageous for these fish in their environment can be the same traits as human diseases.
Yes, so that's interesting.
Some of the others are pretty obvious, like the extreme anemia of ice fishes. That it turns out to be a trait that is fine for the ice fish living in a cold, oxygen-rich ocean. But if we're anemic, it's a really difficult situation. You might need to take erythropoietin, which is a stimulator of blood cell maturation. Other examples of the fish traits that are beneficial that might be deleterious in humans include cardiomegaly, a large heart, and some of the ice fishes have very large hearts. Okay, cardiomegaly in humans is a disease. Another example is the Antarctic fish have evolved kidneys that don't have glomeruli. Glomeruli are the filtration organs of the kidney, and these filtration organs are lost in the ice fishes because they need to retain the antifreeze proteins that they're producing in their blood. So here we could study Antarctic ice fishes and other species to learn about what is a deleterious disease in humans. If you have a disease where you're not producing glomeruli, you're not filtering your urine efficiently, and that's a major problem.
Right. And I imagine this was of great interest in the medical community, and you worked, I think, pretty extensively with that kind of research. Is that correct, to bring out the differences?
Yeah, I have worked at Children's Hospital in Boston.
Yes.
Dr. Len Zahn. I helped him establish the zebrafish as a model for human disease. And I have used the zebrafish to investigate, for instance, the unique blood phenotype of the ice fishes.
So it's really quite a contribution, and I'm sure it is a surprise, to people. These are things that are really very striking. The skeleton change that weakens us, and provides survival mechanism for the fish, and the blood is of particular interest because apart from the fact that it's transparent, or the fish is transparent, isn't it? And that that it seems as though it would be the wrong way to survive is to have your blood go anemic. But
yes, well, it seems like it's the wrong way, but in the context of their environment, it actually works pretty well.
And yeah, so you made a real connection there between environment and what we would call health or vitality, I guess, right? What the things were. Then in terms of your other research, because you've gone on for years in Antarctica, were there other things that you have worked on down there that you have really gotten a lot out of?
Well, another area, and this is ongoing, even though I'm retired, I have colleagues that have taken up this project, is development, the development of Antarctic fish embryos in the context of global warming. So we have conducted studies in which we take and use in vitro fertilization to generate embryos of these species. And then we raise several cohorts of the fish, one at the normal or controlled temperature, what they normally experience, and other groups at higher temperatures. And what we are interested in is how the developmental process is affected by increasing temperature. And this could be very important if the fish don't develop well at higher temperatures, and recruitment to the population would be reduced.
Right. Have you just found anything in that respect then, that the gradual, well, actually accelerated warming is impacting the embryos?
We haven't yet. The samples have actually been submitted for sequencing analysis.
Right.
And we hope then afterwards to look at whether there are any perturbations in gene expression that occur at higher temperature, and what the actual phenotype of the fish is, how rapidly do they develop, and so on at higher temperatures. So that's an ongoing area of research.
Very interesting. Seems very appropriate now that they've discovered all those egg nests on the floor of the. Did that make you think, gee, they're doing really well? But you didn't probably know how abundant those nests were, perhaps in the past.
Not at all. In fact, the remarkable background of that particular finding is that the cruise was actually not devoted to biology. It was an oceanographic cruise, and they put down their towed observational gear, pulled it over the floor, and all of a sudden they were in this immense field of icefish nesting area, and so they were just totally blown away.
Yeah.
As you can imagine, and they called up biologists and say, hey, can you help us tell what this fish is?
We'll have plenty of eggs to look. But that was sort of a cheery message, wasn't it? Because you would think that that species would be impacted rather quickly because of their previous adaptation to extreme cold. So, but they seem to be doing well at the moment, but it will be interesting. You'll be publishing then those studies about the embryos and any kind of changes that you note then.
Absolutely. Yes.
Right.
That's work in progress.
And will you, now that you have retired as an academic, but will you continue your research in any case in the Antarctic?
It's hard to tell what whether I'll be able to do that or not because of the impact of COVID.
Yeah.
The National Science Foundation has very wisely reduced the number of people that they're deploying to our stations. So Palmer Station actually doesn't have any scientists at it at
Ah,
the moment. And there are some scientists working at McMurdo. But the critical issue here is that, if you have a COVID outbreak in a
you're
station
stuck.
with limited medical resources
Yeah.
and is very isolated, you could have a major problem.
Exactly. Like being on the moon, really. Very hard
Almost, yeah.
to treat that because of the storms and so forth down there. You can't always get in and out, I suppose. That is really hard on research, but you just see it across research across the board this year, the last 2 years. It's
Right.
really hurting scientists, it's for sure. But I'm sort of hoping you'll keep up that work. I mean, I'm sure that you look forward to that frigid climate every year.
Yeah, I would like to. I would like to return, but
Yes,
it's unclear at the moment whether that's in my future.
right. But you're going to go and visit your island, right?
I would like to.
Okay. Dr. Detrich, thank you so much for this wonderful information. And I wish you the very best, whether you continue the research or not. You've made huge contributions. Thank you so much for joining us today.
Well, thank you very much, Yvonne, for having me, and I look forward to seeing the program.
Yes, soon. Bye-bye.
Bye-bye.